And now we're going to move on and talk about genes and transcripts. So Leanne gave a really nice introduction to the different annotation methods that we have, but I'm just going to dig in a bit deeper and talk about the two different independent annotation methods. So, but to start with, let's just look at the genes and transcripts that you would have seen on the location tab when looking through the Ensembl genome browser. So on my slide, I'm showing you the region in detail view that you would have explored whilst you were doing the exercises or when I showed you the demo. On this slide, we have different transcripts of a gene, and a gene is a collection of transcripts in Ensembl. So it's absolutely normal for many alternative transcripts to be transcribed from a single locus. They're also known as isoforms or splice variants. And personally, we're not a fan of the term splice variants, as that would imply that alternative splicing is the sole reason for alternative transcripts. However, it is more complicated than that. There are alternative transcription start sites and end sites. There are alternative translation initiation sites, and so on. And all of these transcripts are valid and real. So they could be expressed differently. They could serve different biological functions. So if we focus at the transcript on the slide, the transcript biotypes are color-coded. So you'll see that all of these transcripts have sort of different colors. And we can distinguish between protein coding genes. So these are genes which produce proteins and are translated, and then non-coding genes, which do not result in a protein. So for example, like RNA genes. So a protein coding gene can have protein coding transcripts, which are translated, and those are shown in red. But protein coding genes might also have non-coding transcripts, which do not get translated. So these could, example, be retained intron transcripts or nonsense mediated decay. So these could be fragmentary. They could be missing the translation start sites, so they're not translated, and they might be faulty or degraded before they get translated. And these non-coding transcripts of coding genes are shown in blue. And finally, we also have golden transcripts. So these are called merged transcripts, and they have the golden color, which indicates higher quality and confidence of their annotation. And the golden ones can be coding, or they could be non-coding. So what else can we learn from this image? We have exons. So exons will be shown as boxes, and empty exons or empty boxes are non-coding, which means that they are not translated. They're only transcribed. Therefore, transcripts of a non-coding gene, for example, like you see over here, is going to comprise of empty boxes or empty exons. And filled or colored exons are coding. So you can see that the coding transcript is going to have these filled boxes. However, a coding transcript can also have empty boxes at either 5' or 3' end, and these are called UTRs. So these are untranslated regions. So it's important to remember that not exons are coding. Not all exons are coding. UTRs are exonic as they remain in the transcript after splicing, but they are non-coding as they are not translated. Introns are shown as lines that connect the exons with the boxes. So we see introns over here. And there is a few hints that indicate the gene orientation, so the strand on which the gene is located. The first hint is that the relative gene position of the genome assembly. So you will notice in some cases that you will have a gene track above the reference genome. So our reference genome is this line, this blue line of different shades of blue. So you'll notice that we'll have a gene track above the reference genome and also a gene track below the reference genome. So the transcripts that are shown, or any of the features that are shown above the reference genome, are forward-stranded, whereas the transcripts that are shown below the reference genome are reverse-stranded genes. Another hint is that introns of a forward-stranded genes is going to have this hill-like structure. So it's going to be pointing upwards, whereas the intron of a reverse-stranded transcript is going to have this valley-like structure. So it's going to be pointing downwards. Another hint is also with the gene names. So next to the gene name or the transcript name, you'll always have an arrow that will indicate the orientation. So we use two different independent annotation methods, and both of them rely on real biological experimental data. The first is called the Ensembl automatic annotation, which is applied to every single vertebrate in Ensembl, and the other one is called Havana manual annotation, which is done by a group of annotators. So it's done by real people, and it's only done for human and mouse, and then partially for zebrafish and rat. The Ensembl automatic annotation is a genome-wide gene prediction that uses our automated pipeline. So it's based on real biological experimental data. For this, we take protein, transcript, cDNA, RNA sequencing data. We plot them onto the genome using a sequencing matching technique. So people sometimes think that all we do is that we screen for genomes for open reading frames, but we need to have real evidence that something is translated or transcribed to make that gene annotation. And it takes about a week or two to annotate a genome using our automatic pipeline. And here we have source of biological evidence that we may use. So we take nucleotide data from International Nucleotide Sequencing Database collaboration. So this is a collaboration between 3 institutions, including NCBI's Gene Bank, DNA Data Bank of Japan, and also ENA, so European Nucleotide Archive. We take most of our proteomics data from UniProt, that includes manually curated proteins from SwissProt and unreviewed translations from Tremble. And then sometimes for rare cases, we might also annotate less studied or sampled organisms based on similarity to a better sampled organism. So for example, if there isn't enough evidence or if there isn't enough experimental data for a certain species, then we might use data from a closely related species. So we might be able to predict genes in chimpanzee by mapping transcripts and proteins from human onto the chimp genome. But where we apply this annotation method, we always mention it on the Ensembl pages. Then we have manual annotation. So this is the Havana annotation, and it's also genome-wide gene prediction on a case-by-case basis. It's done by a person, so an annotator who moves along the genome utilizing the different types of data, including databases and papers. So it is more laborious. It takes about half a day to annotate a gene, and it might take up to several years to annotate a genome. And this is why currently it's only done for human and mouse, and then for some difficult regions, it's also done for rat and zebrafish. The annotator can utilize a wider selection of evidence, including the RNA sequencing data for both short and long reads, and also data supporting the transcript structure like CAGE and poly-A sequencing, which can tell us something about the transcription start and end site. But then we also have mass spectrometry protein data that can be used, and also publications. The advantage of manual annotation is that it is more comprehensive, especially for some difficult and tricky to annotate non-coding features. It results in more genes in general, and also more transcripts per gene and more biotypes, especially for those difficult non-coding biotypes like pseudogenes or nonsense-mediated decays. It requires less evidence, so sometimes it could be a bit more sensitive. So if there is small evidence available, the automatic pipeline might skip that evidence without making any annotation. But the annotator can explore further. They can utilize other data types and can make an informed decision based on case-by-case basis. So now that we know that we have these two different annotation methods, this brings me to the golden transcripts, so merged transcripts. So these are, so the gene annotation for human and mouse is a product. So it's an output of the two independent annotation pipelines. So we take these two lists of genes that are produced independently by the automatic annotation and the Havana annotation, and we merge them. And if there are gene models that are agreed upon by these two independently and by these independent methods, then the two methods that come up with a transcript model that is identical, we would give that transcript the golden color, which would indicate its high confidence and quality. So then we have Ensembl canonical transcripts. So this is, if we talk about transcript quality, the Ensembl canonical transcript is a single representative transcript that is identified at every locus. So these are basically transcripts which are on balance. They are most highly conserved, most highly expressed. They are in concordant with other transcript scoring algorithms, and they are also in concordant with UniProt canonical isoforms, and they have the longest coding sequence. And they cover the largest number of clinically important variants, and they have to be complete. And I just wanted to mention here about Ensembl stable IDs. So all of our features have an assigned unique Ensembl stable ID, and these are composed of the ENS prefix, which stands for Ensembl, and then we'll have a letter indicating the type of the feature. So it would be G for gene, T for transcript, P for peptide, and E for exon, and so on. And then we also have Ensembl stable ID for gene trees and transcription factor binding motifs as well. So the stable ID has an 11-digit unique number. For non-human species, there's also a suffix added indicating the species. So for example, we have SMA for turbot and then DAR for Daniarerio zebrafish. And you can find the full list or complete information by going to the link on my slide. And the last thing that I want to mention in my presentation is the gene ontology. So human language is ambiguous. There are many synonyms and multiple terms for the same thing. And we might all be describing exactly the same thing, but using different language. It's the same in the scientific community. So for example, one person could be describing innate immune response, calling it innate immunity, and then another person might be calling it non-specific immunity. And we know that they mean exactly the same thing. However, the computer searches are not going to cope with this very well. And this is why the scientific community has come up with gene ontology terms. So these are basically standardized language, which is easily searchable, and it's controlled to talk about gene function and to make sure that we're all talking about the same thing. And all of these terms have their accession number, they have a name, they have a type, and they will have a definition. Gene ontology terms can be organized in hierarchy, which means that a given term, so for example, innate immune response can have a more general parent term, and then it could also have some child terms, and there could be quite a few of them, just like you see on my slide. And you can find further information on the Ensembl gene annotation system by referring to the publication. Okay, so this leads me to my demonstration now. We're going to be looking at the ESPN gene in Atlantic salmon, and we're going to find out more information about this gene and its transcripts. So I'm going to jump out of my presentation, and we will go to Ensembl browser. And I'm going to use this search option in the center of the page. And from here, I'm going to choose Atlantic salmon as my species. And now I'm going to type in the gene name. So it is important to specify the species that you're interested in, because the ESPN gene can also be found in mice and other species. Once we've specified our species and our gene name, we can now click on Go. So this brings me to the search results. So you'll see that we have some search results over here. We have the ESPN Atlantic salmon gene, and then we also have some search results for the transcripts. You can always restrict your search result by choosing this restrict category on the left-hand side. So you can restrict it to gene, transcript, or gene tree, and so on. I'm going to click on this first search result, which is ESPN Atlantic salmon gene. And this is going to take me to the gene tab. So we've explored the location tab previously, and now we're going to explore the gene tab. You'll notice that the location tab also appears next to the gene tab automatically, so you can always toggle between the two if you'd like. So the gene tab is going to have the gene name followed by the Ensembl stable ID. We're going to have a description for this gene, location. So the location for this is chromosome 15, and then we have the coordinates over here, and the gene is available on the reverse strand. Then we also have some information on the assembly. Then we have further information about this gene, that this gene has 2 transcripts, 189 orthologs, and it has 3 paralogs. And then we have a table for the transcripts. So you can hide this table, or you can show this table by clicking on this blue button. So we have the 2 transcripts over here. And in the first column, we have the transcript ID. Then we have the name of the transcript, the base pair length, the amino acid length. We have the biotype. So these are protein coding, both of them. And then we have the flag for this transcript. So as I mentioned, that we have Ensembl canonical, which is the most representative transcript for that gene. So this Ensembl canonical flag is available for this first transcript. So this is the most representative transcript of this gene. You can always hover over these flags, and it's going to give you a nice description of what Ensembl canonical means. I'm going to hide this transcript table for now. Then we have a summary on this ESPN gene. So we have the Ensembl version, the gene type, and also the annotation method. So this annotation was produced by the Ensembl gene build method. So that is the automatic annotation. And then we have the transcripts shown in the region context as well. On the left-hand side, you'll notice that we have these gene-based displays. So just like we had location-based displays on the location tab, over here we have some different ways to explore the gene. So firstly, I'm going to click on sequence to view the sequence of this ESPN gene. So this is on the left-hand side. I'm going to click on sequence. And we have our sequence over here. So you are able to download this sequence. You can also blast this sequence by clicking on this blue button. And then we have a legend for this sequence as well. So all of the exons will have this peach highlighting, whereas the exons of the ESPN gene are going to have the peach highlighting as well as the brown lettering, just like you see over here. So the sequence is shown in the FASTA format. So we know that because it starts with this greater than sign. We have some assembly information, we have the coordinates, and then we have -1 that denotes that this is on the reverse strand. You also have the option to select a specific sequence and then blast only that selected sequence. So you can just click and drag your mouse across and select a specific sequence to blast. You can configure this page further. So if I go to configure this page on the left-hand side, we can choose the 5' flanking sequence, the 3' flanking sequence. We can choose the number of base pairs that we would like to appear per row. You can have some additional exons to display, the orientation, and you can also choose whether or not you would like to show variance. This is selected to yes for now, but I'm just going to select no. We will talk more about variation data tomorrow. So as you've seen that there are a lot of options that you can choose to configure your sequence. I'm not going to change anything for now. So let's just jump out of this menu. Also on the left-hand side, you'll see that we have some displays for comparative genomics over here. So we will talk about comparative genomics tomorrow. And then we also have information on ontologies. So we have gene ontology. I'm going to click on molecular function. So this is on the left-hand side under ontologies. And you'll see that we have a table over here for gene ontology, molecular function. We have the accession number, the term, evidence, annotation source if available, and then also the transcript IDs. So we have the transcript IDs over here because gene ontology terms are associated with transcripts rather than genes. Then also on the left-hand side, we have phenotypes. We have some variation data, which we will cover tomorrow. And you can also click on external references. So if I just click on this, it's going to then give you a table which will show you the external database IDs for the gene, for our ASBN gene. So we have data, IDs over here from NCBI, projected ZVIN, and also Wiki gene. I'm going to show my transcript table, and I'm going to click on this first transcript ID. And this is going to take me to the transcript tab. So you'll see that we're now on the transcript tab. So I've just clicked on this transcript ID, which takes me to the transcript tab. So we have similar information over here, like we did for the gene. So we have the description, the location, and then we also have some information about this transcript, that it has 14 exons and it is annotated with 53 domains and features. So this is some protein information that we have on this transcript. Then underneath, we have a nice graphical representation of the transcript. So you'll see that we have the introns and exons. So introns are these lines, whereas exons are these boxes that are shown. So we'll have 14 exons on this transcript. We have some statistics information. So over here, it shows us that this transcript has 14 exons and all 14 of them are coding. We have the transcript length over here, and then we also have the translation length. On the left-hand side, we have some transcript-based displays. So we have sequence information over here, and in this instance, the sequence is divided into 3. So we have exon sequences, we can view cDNA sequence, and we can also view protein sequence. I'm going to click on exons. So on the left-hand side, under sequences, I would like to view the exon sequences. And here we have a table that gives us information about the exons. So the exons are ranked over here in the first column. Then we will have the exon stable ID in the next column. We have the start position, the end position. We have the length of these exons. And then in the last column, we have the sequence for these exons. So again, the sequence is color coded. So orange is UTR. Blue will be the translated sequence. And this is also mentioned just over here above the table in the legend. You can also download this sequence. So if I click on download sequence, so it's this blue button on the top of the table. You can download your sequence as FASTA or as rich text format, which is Word compatible. You can have some settings. So you can either select or unselect 5' UTR, 3' UTR, exon, intron, or have the genomic sequence only. And you can also select the size of your flanking sequence. And then we have the guide to the file formats over here. So this is what the FASTA format is going to look like. And this is what the RTF is going to look like. So that's going to appear with all the marked up sequence or any coloration and decoration. So let's jump out of this menu. And then you also have the option to select a specific region and blast that selected sequence. Now we're going to look at the cDNA sequence. So also on the left-hand side, I'm going to click on cDNA. And here we have our cDNA sequence. So this cDNA sequence is shown in 3 lines. And the first line, so the first row, is our actual cDNA. So this is the spliced transcript with UTRs that would be present, if available, that are going to be shown in a yellow highlight. The second row is our coding sequence. So this is our CDS, and you'll notice that it's going to start with ATG. So just over here, it starts with ATG, and then our coding sequence starts with ATG as well. So obviously we don't have any non-coding exons in this specific transcript. So the sequence starts with ATG. And the third row, which is just over here, is actually our protein sequence. And this is starting with a methionine. So you'll see this M over here. Similarly, you can download this sequence, you can blast this sequence, and then you also have the option, if you would like to find out more information about this sequence, you can always click on this question mark icon. That's going to explain you what the different lines are. So the first line, second line, and third line, and also the different coloring on the sequence as well. The next thing that we're going to look at is the external references over here on the left-hand side. So you'll notice that we have external references that are divided into two. So we have general identifiers, and if available, we will also have oligoprobes. So it's not available for this transcript. So I'm going to click on general identifiers. And again, this is going to show you a table giving you the IDs that correspond, the IDs from external databases that correspond to our transcript. So we have information over here from projected ZFIN and also RefSeq mRNA and RefSeq peptide predicted. The last thing that I would like to bring your attention to is protein information. So on the left-hand side, you'll notice that we have some protein information. So proteins are not features in Ensembl, so they do not have their own dedicated tab, as they do not map to the genome directly. They map to the genome via transcript objects. So this is why you can find a bit of protein information within the transcript tab, including protein summary, domains and features, and also some 3D structures if available. I'm going to click on protein summary. And here we have the N-terminus, here we have the protein summary, so which will show you a graphical representation of this translation. So we have the N-terminus of this translation, and here we have the C-terminus. This translation is colored to show you exon boundaries. So you'll notice that we have this pink color as the first exon, and then the purple color would indicate the second exon, and then pink again, and so on. We then have some protein predicted domains that are predicted by different methods. So these are all in silico predictions, not experimentally validated. These are different methods that do not always agree, but if a given protein domain or motif are independently predicted by various methods, then it's most likely to be real. You can also find all of the same information in a table format if you click on domains and features on the left-hand side. And here we have very similar information given in a table format. So we have the domain source, start and end position, the description, the accession, and also interpro information as well. So this is everything that I wanted to show you on the gene tab and the transcript tab. So this is quite a lot of information that you can pull from these two tabs. We're now going to have some time for exercises. So we're going to focus on the exercises for genes and transcripts. And we have two exercises over here. And one of them is in Common Carp and the other one is in Turbo. So I would encourage you to try both of them. And if there are any questions, I'm here to go through the exercises on the screen. So let's say that we will have about 20 minutes for the exercises, and then I'll come back and I'll go through one of the exercises on the screen.